It turns out that these leftovers of glycolysis, there's still some electrons in them that can be squeezed out. And it turns out that in order to squeeze out the remaining electrons in these leftovers, they have to go enter the Krebs cycle. And basically during the Krebs cycle, one of the major things that happens, again, is that the electrons that are found now within these pyruvic acids are squeezed out. They can be released. And again, the electrons, they don't go as naked electrons towards the oxygen. Instead, there's a molecule that picks them up and delivers them to the oxygen. Basically what those NAD pluses do is that they make sure that the electrons get to the oxygen and that the electrons don't react with anything else along the way. So one last thing about the Krebs cycle actually is that not only are electrons released from these pyruvates during the Krebs cycle, but a little bit of ATP. So some ATP molecules are also produced during the Krebs cycle. The thing that happens then to all these NADHs, again, these have the electrons that end up being delivered to where the oxygen is in the cell. So these guys are going to take their electron cargo to wherever the oxygen is. You might remember that the electrons don't go directly to the oxygen molecule inside of the cell. Instead, they go through, or they go to the oxygen via a series of molecules called an electron transport chain. And you might remember that the electron transport chain, these molecules that make it up, they're located within the membrane of the cell. So if you're talking about a eukaryotic cell, this electron transport chain, remember, is located within the membrane of the mitochondrion. But we're going to end up focusing on bacteria, prokaryotic cells. And so in prokaryotic cells, the electron transport chain is found just within the plasma membrane. Right? Because remember, bacteria, they don't have mitochondria. Right? Prokaryotes don't have huge organelles. So again, what happens is that the electrons that are being carried by these NADH molecules, they're delivered towards the oxygen via this electron transport chain. So we'll show you that next. So that NADH, remember, the electron is actually locked up in this hydrogen atom. The electron within that hydrogen atom is what ends up going through the electron transport chain. The electron can either be delivered to complex 2 or complex 1, depending on what type of molecule is delivering the electron. If it's NADH, the electron that's within this hydrogen ends up going to complex 1. Once it's there, basically it gets sucked towards where the oxygen is. And a whole bunch of things happen then that result in the production of a whole bunch of ATPs. Right? So again, the energy that is generated by having that electron move, that energy is used to make something else that has a lot of energy, ATP. So generally this is what happens whenever a cell uses organic molecules as its electron source and oxygen as its final electron acceptor. That's generally how the ATPs are produced. And again, this should be review, right? So you should have talked about all the details of this during Biology 211. And again, this will be our kind of jumping off point for trying to understand how prokaryotic cells do the different types of metabolism that they can do. So in order to sum up what we just showed you here, how about if we do this? So whenever you're talking about an aerobically respirating chemoorganotroph, one of the first things that occurs is that glucose gets broken down via glycolysis. This results in the release of electrons. Those released electrons then are picked up by NAD+. After that happens, it becomes NADH, and that's the molecule that actually brings the electrons towards the electron transport chain. Once it's at the electron transport chain, the oxygen at the end ends up pulling the electrons through. Then the energy of the moving electrons is used to generate ATP.